to all of you who have joined for this wonderful courses. And so we are in the penultimate course. And so before we actually move on to what would be spoken today, so it's like we all have been reading on the newspapers, the headlines, social media about the recent Air India crash, which has taken the life of 270 plus people have died. It's a very sad thing to know, to see, and a live footage of a plane being taken off, being taken, departing from the airport and directly crashing into a building. It's a trauma. Psychologically, it has an impact on everyone. But obviously, a lot of answers, a lot of questions are there. A lot of answers have to be given. But the most prime importance is to know, like, who all, like, the people, the 241 people who have died, or the 270 people who have died in this whole crash. They have to be identified. So as we know from the start, from 12th June, so as we got the information, the whole the labs were started identifying the victims by matching the DNA. So as of now, 3 labs, 40 scientists, 200 police personnel, 300 samples plus from the crash site, because what we see is only 270 on the paper, but obviously we all know that it's beyond 270. Then the samples from the relatives, and as of now, 208 plus DNA matches have been confirmed, and 172 for now bodies or mortal remains handed over. These remains, these bodies, you can't even identify them. You may see them in the coffin, but once you open the coffin, you see nothing. They are all charred. But how has this been done, and how the process is being done is what we're going to understand now. But before this plane crash, also we have had disasters in India. Last year, we had in 2024 the Wayanad landslides, where 36 bodies identified via DNA analysis. Even the tsunami that we had in 2000s, even till date, there are victims unidentified, and it's in the process, and the bodies are in the process of identification too. So when any disaster happens, when any natural calamity or disaster happens, so the process of DNA identification is a very tedious procedure, and it's psychological. It has a huge impact. But as we all have to move on, I mean, no, but on a day-to-day basis, we are much more glued to a lot of forensic shows, which we see on different OTT platforms. You call anything, you take the English web series or the Hindi web series or whatever. So we are all glued to it, and we understand the kind of a crime happens, how does a murder take place, and how is the suspect identified in the end. So what we today are going to talk about is forensic science, and how you can build a career out of it, and with the help and being a forensic scientist, what all cases can be solved and what innovations are there till date. If 250 plus matches have been done, then it is all because of technological innovation. And in 2025, the technology has innovated to a larger scale beyond our capacity too. So this is what is going to be my presentation. So I would like to give a brief introduction about me. I am Dr. Arunagiri Subramanian from HiMedia Laboratories. So I work in the molecular biology department, and it's not that if you look at my background, I'm actually a medical microbiologist. I've done my PhD in medical microbiology. But as I joined HiMedia, forensics, I have no forensics background in that way, but studies, as I go through a lot of literatures, understanding what exactly is this. So this forensic science has fascinated me, and today is going to be a very interesting aspect of what forensics is, and what you can become and what all you can do. So let's understand the definition of forensic science. It's actually originated from the Latin word forensis, meaning public or suitable for court. Because if you see, if you take any case, end of the day, the verdict is produced by the court. So that's how the word forensic is coined. So what is its objective? The objective is to collect all the evidence which can aid in criminal investigations and legal proceedings. So that's a very basic understanding of what forensic science is all about. But today, if you see, forensics has developed into multiple disciplinaries. You start from DNA analysis, you go to blood stain pattern analysis. If you all had watched Rudra, the web series, you will see, how the blood pattern analysis is considered for identifying the crime. Medicinal chemistry, toxicology, forensics archaeology, then forensics geologist, odontology, forensic engineering, ballistics. Ballistics is important. You know why? If a bullet has been fired from a gun, it's important to understand or to see the match the bullet and the gun. So it has its own way of working. Digital forensics, obviously, cybers, crime, scams, a lot. We have seen digital arrests and all those things. So digital forensics come into play, and forensics entomology, forensics anthropology. So forensics has divided itself into multiple small divisions. This is just something that we read, but there are more divisions that are being protruding out nowadays. To understand what exactly a forensic scientist do is, there are three steps that they have three main objectives. To analyze. They have to analyze the evidence that they have with them. So the evidence could be from a crime scene. The evidence could be from any particular thing. So their objective is to understand the evidence. Second, based upon the evidence, they have to prepare a report, and that report is submitted in the court. Okay. And that it could be, so the evidence could be from the areas of DNA, toxicology, ballistics, and more, and as I put in the earlier slide. So to become a forensic scientist, now let's talk about in India. It was not till the Nirbhaya case that we never knew what forensics was all about. There were sexual assault cases happening in India, but the Nirbhaya case changed the way forensics was to be seen in India. If you see the Western countries, forensics have grown a lot. But in India, now in 2025, it has grown huge. But earlier, when the Nirbhaya case happened, that was a changing point. And then the government started thinking, there has to be something has to be done. As we have doctors, as we have physicians, as we have chemists, as we have engineers, there has to be forensic scientists too. So then came the nurturing of universities and all other things. So what one needs to become a forensic scientist? You need to have a bachelor's degree in biology, chemistry, or a related science. And then you do an advanced roles require MSc, obviously master's, and then you can follow by doing PhD. It may so happen you may do a master's in biology or zoology, and then you can do a PhD in forensics. Now, the courses, the kind of disciplines have become very open in our country. But what is important, you need to have a good observation skills, because thoughtful process is important here. Analytical thinking, a good way of report writing, because every evidence count, even a small evidence that may not be really you may think, oh, this is not really important right now, but it has still to be counted and written on the report. And obviously courtroom communication, because to become a forensic scientist, like if you see, the forensic scientists have to visit the courts, because when cases are being heard, the forensic scientists have to go to the court and defend their proceedings. They have defend the way they have done the analysis of that particular crime, of the particular crime. They have to say, okay, why is the suspect and or why is the suspect not the suspect in that way. Let's talk about the DNA matching and all that we talk about. But let's understand a brief history into the forensic science. So every contact leaves a trace. Yes, today, wherever you are sitting right now and listening to this particular seminar or to this course, you may be in a cafe, you may be at your home, or you may be at your friend's house. You may not know, but you would leave a trace of your at that particular place. It could be a hair, it could be a nail, it could be skin scrapings, or it could be even the coffee cup that you're using it. Even the fingerprint touch on the coffee cup could be an evidence that you can be caught in that way also. So every contact leaves a trace. That's been coined by Edmund Locard. So right from 441 BC to AD 1247, forensics has grown over the period of time. And in AD 1794, the first gun crime solved by forensic test. Then we have microscopic analysis. Yes, it's important. If you all talk about DNA, microscopic analysis is also important to identify the microbial way or spermatology analysis, or there are multiple things to analyze using a microscope. Then it was in AD 1923, the first US forensic laboratory was established. But what is important is the background of to identify these forensic investigation is all we talk about nowadays. So if in 1866, Gregor Mendel came up with the pattern inheritance, followed by Frederick Milner, who set out to identify the nuclein, and which we now know as a deoxyribonucleic acid, DNA. And Oswald Avery identified the DNA as a transforming principle. It was in Erwin Chargaff in 1950 who discovered that DNA composition is species specific, and Rosalind Franklin, who photographed the crystallized DNA fibers. Not, as we all know, 1953, the Watson and the Crick model, who discovered the double helix structure of DNA. And in 1962, Marshall Nirenberg, the first person to sequence the bases in each codon. So the process of identifying the DNA as the base for forensics was done for centuries, and now in the 21st century, we all know that the DNA forms the base for forensic identification. As we saw in this crash, the DNA is the base for forensic identification. So I don't want to really go into the depth of the DNA. We had courses of it. So we all know that the nucleotides are classified as pyrimidines and purines. The purines are two-ring structure. The pyrimid has one ring structure. So where exactly is the DNA been seen? So the DNA is generally has been observed or be found in the nucleus of the cell, and is packaged into chromosomes. As we all know, 22 autosome chromosomes and 1 reproductive chromosomes. And where all you can recover the DNA, you can recover the DNA from any part of the body. But how forensic came into effect, or how the birth of forensic DNA testing was by Professor Alec Jeffreys. So he was a person who identified a concept called as short tandem repeats, or STRs. So he just was going through an analysis, and he found that there are some kind of 2 to 6 base pair repeats which are there in the body, and they are repeating itself. It could be a 4 repeat, it could be a 6 repeat. So these repeats in the body cause or coin the uniqueness of the person. So what are STRs actually? They are tandem repeat sequences, short sequences repeated multiple times. They vary between individuals. That's why we are all unique. Even we are unique in every way. So making each person's DNA profile unique. The analysis of this variation has led to the foundation of DNA fingerprinting, and now a central method to forensic identification. When was this first method employed of using repeats as a process of identification? It was in the year 1986 to investigate a 15-year-old Don Ashworth rape case murder. So DNA as such for identification of the suspect was first utilized in the year 1986. Okay, so we all understood what repeats are. So there are small repeats, and which bring about the uniqueness of a person. But where are these STRs located? So they are all throughout the human genome. So as we said, the 23 chromosomes, 22 autosomal chromosomes, and the two reproductive or the one set of reproductive chromosomes. So the STRs are all part of these 23 chromosomes. And these STRs, and there are multiple STRs, multiple repeat patterns in the body. And it is very vast to understand these things. Right now, I don't want to dwell into these things because it's a subject to understand different way. So these STRs are part of the human genome, including the one autosomal chromosomes and the reproductive chromosomes. How are these STRs classified? So the STRs are classified into multiple ways. It could be a 1 repeat, or it could be a 2 repeat, 3 repeat, 4, 5, 6, n number of repeats. It could be from 1 repeat to 16 repeats to 18 repeats and all multiple repeats. So these classification of the repeats bring about the classification of the STRs and thus the uniqueness in the particular person. But how do we identify these repeats? It's important, like how do we go in identification of these repeats? So we are all glued to this crime, CID. दया कुछ तो गड़बड़ है, किधर ना किधर तो हमें ढूंढना पड़ेगा ये रिपीट कहाँ से मिलेगा. So how do we identify these repeats? Obviously, samples, all the biological samples is the main base to identify these repeats. So what kind of samples can be utilized for identifying these short tandem repeats? It could be blood-stained cloth. It could be fabric. Okay. It could be sexual assault samples. Recently, also as I said, in India, there is a lot of sexual assault samples that happens. Bone and teeth. What we saw in the Air India crash, the bone and the teeth are the main biological samples which have been used for identification. Tissue, because in India you find a lot of abortion cases. Okay, so tissue is one sample, is one way of identifying the DNA. Fingernail scrapings, cups, bottles, cigarettes, toothbrush, hairbrush, all your cosmetics that you use, eyeglasses, things which are used for intimate affairs, tape, literature, and stamps or envelopes. These are just samples that have been described here, but beyond this, there are more samples also that from which the DNA can be identified and the short-term repeats can be identified. So just to talk about this image is to collect a blood swab from a crime scene. So you would see a lot of blood being spilled on the ground, on the wall. So the crime expert, he comes and he takes the swab. With the help of a swab, he takes the blood, the dried blood, and that goes to the laboratory. Cigarette butts. When we smoke, we tend to throw the cigarette down. So that is an evidence because the saliva goes into the cigarette, and from that tip of the cigarette, end of the cigarette, we can actually extract the DNA out. Hammer. Multiple, you could be a sword, it could be a hammer, it could be a rock, anything. So these are all also where the DNA can be identified for. Bones, obviously, burnt bones or bones from the buried sites, so where the DNA can be identified too. So like these, there are samples from where the DNA can be identified. But what is the process of identification of the DNA? Like, how do we go about it? There are steps involved in the process of the DNA identification. Now, before we really move on to the reality of it, let's understand this case. So we have a case where we have to compare the DNA collected from the crime scene to determine which of these three suspects was present. So if anyone could actually say, from the crime scene. So there are DNA bands from the victim and of the crime scene, and there are three suspects. So if anyone from the audience would say who is actually the suspect from the three.
So suspect.
Say who's the suspect from the three. Yeah, so good. Multiple answers are now coming in, and suspect 2. You're right. The suspect 2. is the main suspect who has brought about the person who's involved in the crime. Now compare the profiles of three men with that of the mother and the child to determine the biological father. So now who could anyone say who is actually the biological father of the child? So just compare the DNA profile of the mother, the child, and of the fathers. Okay, let's see the answer. Yes, dat3 is the right answer, and dat3 is the biological father of the child. So you just see the match of the mother, the DNA bands of the mother, and that of the dat3 with the child. So he is the biological father of the child. But how do we get to know these DNA? So how do we go about in the process? So what exactly is the process for identification of the DNA or identification of the short tandem repeats? So there is a workflow for it. So the first step is obviously you collect the sample from the crime site, and after the collection of the sample, it goes to the lab. So the first step is to do the DNA extraction. So you had a course on DNA extraction, so there are multiple processes involved in DNA extraction. And after you have the DNA extracted, then you quantify the DNA. Now, if you look at here, I have put an image of a real-time PCR machine here. And but in general, we do quantification using the spectrophotometer. There is a slight difference in the quantification of a DNA done using a spectrophotometer and done by the real-time PCR. If you do the spectrophotometer analysis, you actually do the quantification of the whole genomic DNA. But here, we are very specific to the human DNA quantification. Then the next step is you do a sequencing, but before the sequencing, you do a PCR, conventional PCR of multiple markers. Now, if you had seen the 22 chromosomes image, now let me go back to that slide. Yeah, if you look at this particular slide, we see 22 chromosomes, okay, and 22 and 24 total. So each chromosome is having one or two markers. So when we do a multiplex PCR, it's a multiplex PCR of 25 or 27 markers. So that's the amount of markers that we are going to consider for DNA identification. So hence, these are very important steps to understand. So in the PCR, after our quantification, we do a PCR for 27, 25 markers. And these 25 markers, the PCR product is then put into the sequencing machine. And after the sequencing is done, we do a bioinformatic analysis of it. So obviously, we all spoke about DNA extraction in the previous courses. PCR also has been spoken in the previous courses. But why DNA quantification, and that too by PCR? So as I said, when you work with forensic samples, all these samples are not good samples. They are all very degraded form samples. Like example, like this crash, Air India crash. All the samples are all burnt bones, charred bones. So you are not going to get a good DNA from these charred bones. Hence, quantification by PCR is an important tool to understand what is the quantity of the DNA from that particular sample. So the quantity of the sample could range from 3 picogram per microliter to 300 picogram per microliter. So that's the DNA concentration that is achieved from these kind of forensic samples. It could vary. It could definitely vary. And apart from these DNA concentrations, these samples come up with a lot of inhibitors, like they're called as PCR inhibitors. So that's a different thing. Like what PCR inhibitors are. So what exactly do? They basically obstruct the PCR amplification. So there could be PCR inhibitors, or the DNA could be completely degraded form, or the DNA quantity is very low. These three parameters are not directly identifiable using a spectrophotometer analysis, and hence real-time PCR amplification plays a very crucial role. So once you quantify the DNA, then you do a PCR for 27 markers or 30 markers or how many markers you want. And after the PCR is done, the PCR product is then loaded into the sequencing machine to get the genetic DNA profile. So as you see, the green arrow indicates a DNA concentration of a 0.03 nanogram per microliter, or you see a red arrow which is indicating a DNA concentration of 3 picogram per microliter. Hence, the concentration varies, and hence real-time PCR is very important here. But how do we interpret these STR analysis? Is that by two main terminologies. One is called as the locus, where we find the STRs, which is the specific location on the chromosome where a particular STR is found. And the next word that we are going to see about is alleles. So alleles are of two types. They could be homozygous alleles or heterozygous one. So what are alleles? It's a variant form of a gene and occupies a specific location on a chromosome and can influence the phenotype of an organism. So you have the dominant alleles, or you have the recessive alleles. It could be homozygous, or it could be heterozygous, or it could be either ways. So how are these STR results interpreted? So if you look at this particular image, you see multiple terminologies being here. Allelic peak, RFUs, peak area in RFUs, peak height, allele labels. So these STRs, so let's be very simple. Now, these STRs are which bring about multiple repeats in those. So is coined by the word allele label. So the 15 year is the allele label year, which is part of the particular loci, which is like here the locus label, D3S1358. So this is how you interpret the short tandem repeat pattern of a particular sample. Now let's take an example here. So this is a full DNA profile of a particular sample. And if you see, there are peaks, which are basically the alleles of that particular loci. So these alleles, as said earlier, they could either be a homozygous allele. So what does homozygous allele mean? Means that the alleles are, the homozygous means that the chromosomes are the same from the mother and the father. The alleles are the same from the mother and of the father. So that's why you will have a single allele, and that's called as a homozygous allele. But what is heterozygous allele is that there are two alleles, one coming from the father and one coming from the mother. That's why you will have two alleles of one particular loci. So in your own DNA, you could have some loci which are of a single allele, which is there's homozygous, or they could be two alleles, which is heterozygous in nature. So with this help of a pattern, so you identify the alleles. So these alleles, so if you see the numbers, one allele is having 13, one is 30, one is 10, 11. So what are these numbers? These numbers are nothing but the nucleotide repeats. As I said earlier, the 2 to 6 base pair repeat. So these are the repeats. So 13 repeats, 30 repeats, 10 repeats, or 11 repeats. So these repeats bring about the alleles in the particular loci, and there's a whole process of doing this, but it's just a very simple way of I'm talking here out here. So if you have to match between two single source profiles, so as how in a crime case, so you take the sample from the suspect and you take the sample from the evidence, and then you do the whole process of extraction and you go to the profiling, and you would see the match. You would match the whole profile of the suspect and from the DNA from the evidence, and if there is a complete match. So if there is a match for all the 27 loci, then you will say that the DNA of the suspect is matching with the DNA from the crime evidence, and hence the DNA, the person has done the particular crime. So this is how the match is done in reality. But there are other ways of matching too. Like here, sometimes a match is done from a mixture profile. Now, what does a mixture profile actually means is that in cases of sexual assault, when there are 3 to 5 or 10 people involved in a sexual assault of a sexual assault case, so it could so happen that only 4 or 5 have done the sexual assault, and the rest have just helped in the process of sexual assault. So there are 10 suspects now. The DNA from the 10 suspects are taken. The DNA from the victim is taken, or the DNA from the let's example a sexual assault case where you take the clothes from the female, and then you take the sample, you cut the cloth, because there you have in the cloth you would have the DNA of the male and the female, like example a spermatozoa cell. So after the sexual assault has happened, the sperm is released into the fabric. You take the fabric of the victim and then you cut, and now you would have the DNA of the victim and then of the people who have done the sexual assault. Now, since the sexual assault is done by the male people, so you take the male DNA out of it, and that male DNA would be a mixture of multiple suspects, so 5 suspects. Then you do a complete profile of it, you do a complete match, you take the DNA from these 5 suspects and you match it with this mixture DNA. And now if you see, the mixture evidence sample has got multiple alleles, 15, 17, 18 of one loci, 7 and 9 of another loci, then 11, 12, 14 of the third loci, and 10, 12, 17, 19, 23. But if you see that the STR of the evidence sample is only some alleles are matching, like the 15 of the suspect is matching with 15 of the evidence. So the 9 is matching, 8 and 12 is matching, 12 is matching, and 17, 19. So from this, it is identified that there's only one person involved in the complete sexual assault of that particular case, and the rest is not involved in the case at all. So why STRs are preferred? Because they are highly sensitive. Multiplexing up to 30 markers can be done. It takes 24 hours at least for analysis to happen, and it is a very cost-effective process. But what we see is all about STRs being identified from autosomes, but there are Y chromosome analysis also be done. Like we do Y chromosome analysis for mixture cases. So it focuses on the STR repeats found only on the Y chromosome. And since the Y chromosome is inherited along paternal lines, all male relatives share the same Y-STR profile. And it is also useful in sexual assault cases with multiple male contributors. But the next part that we are going to see is mitochondrial DNA analysis. We all have STRs are generally identifiable from the nuclear DNA. But at times, what happens? The nuclear DNA is either in the very degraded form, and especially when you're analyzing hair shaft, bones, and all bone samples, nuclear DNA may not really play an important role, and hence mitochondrial DNA, which are extra chromosome, they play an important role for analysis. They are less discriminating than the nuclear DNA, and they are valuable for identifying missing persons of historical remains. If you talk about the inheritance pattern, it is the mother who is actually contributing the mitochondrial DNA. The nuclear DNA is contributed by both the father and the mother, but it is the mother who is actually contributing the mitochondrial DNA. So what are the real-world applications of STR analysis? It is used for crime scene investigations, missing person identification, disaster victim, like as we see here now in the Air India crash, paternity testing, and cold case resolutions. So just to talk about what are the new emerging fields in forensics, you have forensics genomics, now obviously next generation sequencing applications. You have forensic proteomics, and then you have microbial forensics. Apart from these, there are environmental forensics, and many disciplines of forensics have evolved nowadays. So what I spoke right now is all about wet lab based work. You do a DNA analysis, or you do a toxicology analysis, or any kind of analysis. But as a manufacturer, it is important that we need to make sure that all the supplies, all the reagents, consumables are all human DNA free. Now, this is something very important for us to understand, because there has been a case that has happened. Now, let's talk about that. Not a very haunting pic, but there was a time where the police were involved in to hunt the woman without a face. The 16-year search for a serial killer who never existed. So what exactly is this? This case is called as the Phantom of Heilbronn. It's a mystery which has baffled European detectives for many years. So what exactly is the mystery? So this Phantom of Heilbronn is often referred to as a woman without a face. It was hypothesized that So there was a female serial killer whose existence was inferred from the DNA evidence. It happened between 1993 to 2009 with 40 in 40 different crime scenes. And the investigation was very big, as you can see here. And what was the final outcome of this particular case is that the investigators discovered that the same DNA sequence on the burned body of a male asylum seeker in France was matching with the DNA from Germany also. And they found that all the reagents and the consumables that they had used to collect the DNA evidence had actually been contaminated by a manufacturer. The woman was involved in the manufacturing of the DNA swab. So the phantom serial killer did not actually exist, and that the laboratory results were due to contamination of the cotton buds used for DNA profiling. So this phantom of Heilbronn brought about an answer. Now we have standards, regulatories, they're all important. So this brought about an answer, a regulatory called ISO 18385. So what is exactly ISO 18385? It's basically the objective is to minimize the risk of human DNA contamination in products used to collect, store, and analyze biological material for forensic purposes. So this is an accreditation that every manufacturer has to acquire before they supply, like we supply forensic kits to forensic labs in India and also abroad. So we make sure that whatever manufacturing that we do is free of human DNA contamination. So the very short way, your journey starts here. This forensic science is very dynamic. As I said, this very short time is very limited for us to expect everything. But I hope you got a brief idea of what exactly forensic science is. It's a dynamic and interdisciplinary and impactful this one. Choose your specialization and develop technical and ethical rigor. Be the link between science and justice. There is National Forensic Science University in India. There are multiple campuses where you can actually enroll yourself for the forensic courses. There are different labs. There are multiple labs. There are, if in India there are 7 central forensic science laboratories, and there are 32 state forensic science laboratories, and there are 200 plus regional science forensic laboratories. So the number of labs in India are huge. Forensics as a subject is very booming in India, and you can choose a subject and build a career, and you can see there's a huge scope in it.